Okay, so Dr. David Tarpy, here it is again.
All right, let's go ahead and finish this up. I'm always really loath to talk right after lunch, because now all your blood is in your stomachs. All right. What the heck am I talking about? Diagnosis. Okay, thank you. That's it. As you can see, I have a couple lectures already prepared, so kind of forgot. There we go. All right. All right, well, thank you again to the Cabarrus County Beekeepers. Great to be here. Thank you again for the truck. I'm sorry I neglected to talk about that, right? But you guys were, and Kim Flodham. I thank Kim Flodham profusely for his very kind donation. It all went to that nice shiny truck out there. All of that came about randomly, where we've kind of grown in our program. The program is not just me. In fact, I'm the least important individual in our program. Jennifer Keller, of course, is the most important person in our program. And it just started with her and me, and then we'd add a student here or there, and now we're at like 23 people in the lab, and we only ever had one truck. So you can imagine how competitive it would get, to get the keys to the truck so people could do their thing. Otherwise, they're using their own personal cars with the smokers hanging out the window. We've all done that, right? So I had just mentioned that one day about how that becomes a real bottleneck in just our day-to-day operations. And Rick Core, just a light went off in his head and kind of challenged all the clubs about, kind of funding something very tangible like a bee truck. And you guys just responded just wonderfully, and we just absolutely love it. Call it Clifford the Big Red Truck. I said that before, right? So I think it's really been great, enable us, especially our students, to do a lot more. So kind of on the heels of what we were talking about before on our queen research, I've recently developed this talk. It's not entirely well polished, so please forgive me for it being still kind of a work in progress. But really, what it comes down to is, because we've found in our research that queens have these problems that we all know about, but we're looking at the queens and we're not finding anything obviously wrong with them, but still we're experiencing these problems. So it's a real kind of head scratcher as to why this is. And so it's really made us doubt and kind of rethink what we mean by queen problems. And I guess I'll give you the punchline, since you're all going to be asleep in like 10 minutes anyway, that the current thinking that we have right now is that the things that we're calling queen problems are usually really more colony problems. That it's really, we're using the colony, we're looking at symptoms of the colony, and then we're blaming the queen. That's kind of how we've been taught. That's a very easy explanation, and we tend to gravitate towards easy explanations, especially when we're the ones not raising the queens. So I think it's hard to tease those things apart, and so this is more of a collective discussion and kind of communal voyage of trying to explore that together, right? And so hopefully you'll look at it in that context. And so just to show that this is a real thing, this is a study we did quite a while ago, 10 plus years ago now, where we're looking at Italian and Russian colonies, and we're looking at their longevity. So that's of the colonies themselves, and then this in the second year, we're actually tracking the queens. We requeened them. And so really, you know, we say that queens used to live 3 to 5 years, and now, you know, they live 1 to 2 years, if that. And so there's actually not a whole lot of data on that, but this is kind of among the closest that we have, where, you know, queens don't live 2 years, period. It's pretty astounding, whereas, you know, they used to live a lot longer. And so I think it really is a problem, because every time that a colony goes through a requeening event, they lose productivity. It's a risky thing. It's not always sure that they're going to get a new queen. That new queen might not be as good as the mother. Like, there's a lot of things that can go wrong in that part of the turnover, the generational turnover. And there's that productivity lost, and it's a big stressor, which can introduce all sorts of other problems. So it's a real problem. I had showed this before about how the mating number of the queen tracks with the survival, because the more genetic diversity within the colony, the more likely they survive. So I won't go through that again. But here's another example of how queen problems interface with colony environment in the real world situation. This is again with Dennis Van Engelsdorp, Jeff Pettis, and Kirsten Trainer, about this one particular study. We were looking at colonies where there is a queen event, and this is where, like, supersedure, or they found a virgin queen, or queen cells, or drone-laying queens, or just something wrong with the queen, as though they were perfectly fine here on the left. And then they looked at this measure called the hazard quotient, or the HQ. And what the HQ is in essence a weighted and additive measure of the pesticides in the colony, in this case in the wax. So if you have in the wax a relatively benign pesticide like a fungicide, right? Fungicides are designed to kill fungi, not insects. So they have a relatively low toxicity, right? So you can have a high level of that in the wax, but a low toxicity, so it doesn't add much to the HQ. Or you could have just a little itty bit of a very highly toxic insecticide, and that adds a lot to the hazard quotient, right? And you add all of those up. You find the dozen or so pesticides that are in wax, and then you add up the weighted risk of each of those compounds, and you come up with this cumulative hazard quotient. And in essence, what it shows is that polluted wax, you tended to have colonies that had queen problems compared to those that didn't have. So there seems to be an association there, right? Again, not sure of cause and effect. There's a lot of other things that go in here, but there's clearly an association of the nest environment and our queen problems. Okay? So I kind of broken it down just very simple terms of just categorizing different things in the environment that could result in having queen problems. So biological problems like the rearing conditions of the queens or pathogens within the colony, chemical issues like the pesticides in the wax or the pheromones that the queen or colony is producing. Our management practices have a lot to do with some of these things, like the stock that we use or our own miticide treatments, those kind of things. Then you have environmental factors, you know, like nutrition, their forage, the intensification of the landscape, all sorts of other things. So there's all sorts of environmental, biological, chemical factors that can come into play here. And so my thinking now is that a different combination of all these different factors result in different queen events, right? Right now, all the surveys and everything that we're saying, oh, it's bad queen, good queen, right? Well, that's a binary thing, but bad queens go bad for different reasons, right? And so here are just some of the things that happens to queens in this catch-all phrase of queen events, right? So one is, we kind of talking about it before, which is just the queen herself, that the the queen is of poor quality or small or inseminated improperly, right? That's like the queen reproductive quality sensu stricto, right? It is the queen, not the environment. But these other things that we as beekeepers bad queens are really more colony level traits, like poor brood pattern, drone layer, supersedure. So poor brood pattern, right? Look at a brood pattern, you see shot brood, you're like, oh, bad queen. Well, who raised those brood? The queen? No, the workers, right? So by definition, that's a colony level trait. So it's not just the queen that resulted in that poor brood pattern. Now, yes, it can be because the queen was improperly inseminated, or she just doesn't know how to lay because she's dumb or something, right? But it's not just up to her. But we tend to associate that with just a bad queen, right? So we'll kind of go through these kind of things. And part of the idea here is to look at different symptoms and try to assess whether this is, a queen's fault or the colony's fault, or whether it's some combination of the two, or neither. We'll start with the neither. Whose fault is this? That's the beekeeper's fault, right? Now, I know nobody's done that. You ever stepped on a queen?
Never.
You ever looking through a hive and you just can't find her, and you've been through every single damn comb like 10 times, and then finally you look on the bottom of your boot? Yeah, it happens. This is the result of a queen being dissected for scientific purposes, so that was our fault. This was a picture from Rick Core, which I absolutely love. You see that? That's a queen, and there's a bee's stinger right in her head. It's kind of weird. Never seen that before. So, sometimes it's obvious, or intentional even, right? But I want to talk about how this queen quality concept is not just binary as a function of good queen, bad queen, that there's lots of things going into it. But the whole idea of the queen and her quality itself is really more of a continuum. And I alluded to this in the previous presentation, where we can intentionally make bad queens, right? By taking old larvae and starting to raise old worker larvae as queens. And so they've already spent a day or two or three developing as a worker. So when they start being raised late as a queen, they're more worker-like, and so they tend to be, intercaste. They tend to be in the middle of a good high-quality queen versus a worker, right? So there's really more of a continuum, a spectrum of femaleness, right, of queenliness within a colony, more than the endpoints. In a normal functioning colony, we'll see the two endpoints. We'll see a really good queen or a worker, but you very rarely see these things in between. And so to underscore that the quality of the queen itself is really important, we can make these intercasts, or these really crappy queens, on purpose, and then put them into colonies. And we did this with Juliana Rangel, who, when she was in our lab, she's now at Texas A&M, my counterpart there, and then of course Jennifer, where we establish colonies with low and high quality queens and measure them for their colony growth over time. And what these graphs kind of show you, I don't need to walk them through, but low quality queens produce low quality colonies. They're smaller, they're not as well mated, the colonies don't grow as fast, and then over time, that is manifest by them not having as big populations, raising as much brood, so on and so forth. So it's important then to have really good high quality queens, and this has been shown empirically. So that's important. So I don't want to dismiss that there's variation in queen quality, and that sometimes it is the queen's fault, right? Sometimes poor queens are just that. But I think so often that we're kind of diagnosing things based on the colony, not the queen herself. So a lot of beekeepers are just looking at the brood pattern and assessing the queen based on that. And so here's what you tend to see, right? You see this shot brood. How many people have seen this in their colonies, right? Is this a queen or a colony problem? Or both? Well, it is both, but in this particular case, this is the queen's problem. How can you tell? You can't. I just know this because this was a genetic mismatch of the queen. I don't know how many people know about sex alleles. Have you ever heard of diploid drones? Okay, so this is how this happens. Genetic lesson. You're really going to go to sleep now. So queens, females, workers and queens, come from fertilized eggs. So they have pairs of chromosomes, right? Males come from unfertilized eggs. They don't have fathers. They only have mothers. So they only have one set of chromosomes that comes from their mother, right? And so on the 11th chromosome, honeybees at 16, right? There's this one gene locus called the sex locus. It's called the sex locus because it determines sex, the sex of the individual, male versus female. By the way, bees don't have gender. They have sex. Anyway, so there's no ambiguity, by the way. So in order to determine femaleness, or this single gene, right, has multiple forms. Just like we have different alleles for our eye color. We have brown, blue, hazel, different forms of that single gene, right? Well, at this sex locus, they have like a dozen or more of these alleles out there. And we'll just number them 1, 2, 3, 4, 5, whatever, right? So when a queen has two different forms of this gene, mates with a drone that has a different form, then when they make workers, they all have workers that have two different copies of this gene, right? That results in viable female workers, and so you get 100% brood viability, like that, right? However, sometimes queens will mate with a drone. She has no way of knowing this, not like they're wearing name tags, but she will mate with a drone that happens to share a copy of one of her two sex alleles, right? So she has an X1 and X2, and he happens to have an X2. So what happens is that half of his offspring, half of his worker offspring, will get two copies of the exact same sex allele. And so femaleness is not from being fertilized or not. Femaleness comes from having two different copies of this sex gene. That's what makes you a female. If you only have one copy because you don't come from a fertilized egg, you become a normal male, a normal drone. But if you have two copies of the same allele, you also become a male. But that's the diploid drone. You're diploid because you came from a fertilized egg, but you don't have two different forms, therefore you become a male. Now these males, these diploid males, are cannibalized in the first day of their larval stage. They hatch from the egg, they smell wrong to the nurse bees, and they cannibalize them. Ergo, you get this half of the brood. She's laying an egg, fertilizing it, thinking it's got to become a worker, but it gets cannibalized, and so 50% of her offspring don't survive. That's shot brood. That's this, okay? So is that the queen's fault? No, it's the damn drone that she mated with, right? But it's her fault because she stored the sperm and didn't ask, hey, do you have the same sex allele as me? She didn't, do her due diligence. So this is the queen's fault, but it's unescapable, right? It's kind of a random chance that she mated with a dud rather than a stud. So, but again, we're using brood viability to make an assessment of a queen, and in this case, it really is her fault, right? So it's just like this, right? Here's bad brood pattern, shot brood. What's this?
That's Varroa, most likely.
Varroa?
Well, it could be about that.
See the sunken cappings? Right? This is American foul brood. Is this the queen's fault? Not really. I mean, maybe you could say that the genetics of the queen and their ability to clean out the brood disease, but no, this is, well, maybe it's the beekeeper's fault, maybe it's your fault for using old comb, right, or something like that. But yeah, this isn't the queen's fault, but I think a lot of beekeepers will look at this, oh, that's shot brood, that's the queen's fault. They requeen it, and the colony still dies. And they're like, what happened? Well, you learn your diseases, right? Like this, what's this? Not shock brood. EFB, exactly, right? So this is, again, you can lead to bad brood patterns because of diseases, not just because of a bad queen. So this is shock brood, right? Mummies, a lot easier to see. You probably can't see in the back anyway, so we'll just fly through these. This is that PMS, right? Or idiopathic brood disease syndrome. IBDS. Really impress your friends by saying that fast. Idiopathic brood disease syndrome. But this is, kind of where the mite loads are so high, or the viral loads that they're vectoring are so high that the brood is just a really bad pattern. So this whole concept of using brood pattern as a proxy for queen quality clearly doesn't always hold up. Right? And so we need to tease that apart. And this was actually tested by Katie Lee, well, now Dr. Katie Lee, who got her PhD with Marla Spivak at University of Minnesota, and then our genetics technician, Aaron McDermott. And so what Katie did, she was working on the Bee Informed Partnership Tech Transfer team in the Midwest. She was working with these large commercial beekeepers. And so she was like, hey, I see a lot of colonies, I see a lot of queen problems. Whenever I find a bad queen, I can take that from this beekeeper, but I'll also convince them to go to the hive next door and take a good queen. And I'll send the pair of queens to you at our NC State clinic, and we'll analyze them for every single thing that we can. The queen reproductive quality, the sperm viability, sperm count, all this other stuff. At the same time, she's going to take a sample of wax, and we tested pesticides. She took a sample of the workers, and we did viral analysis of the entire colony. And we did all of these things in the hopes of finding, in these colonies headed by bad queens, they had this thing, whereas the colonies headed by good queens, they didn't, or vice versa, right? To find, again, a smoking gun. And so what we found was that in some cases, the poor queens, or the ones where the colonies had a bad brood pattern, and therefore had a bad queen, they had really high levels of this one particular virus. In another case, they had higher levels of this one particular pesticide. In another case, they had bad sperm viability. But there was no consistency among these different operations, or from pair of queens to pair of queens. So there was no smoking gun. If you haven't noticed yet, a lot of our research is really frustrating because we go try to find answers and we just come up with more questions. But this is part of it, right? So we weren't finding an easy solution here. So frustrated, and during the 10 months of Katie had to think about this up north, she thought about what to do next year. And rather than doubling down and being equally frustrated, she did something which I consider quite brilliant. She's like, well, why don't we do this? Why don't we take the colony where they had a good brood pattern and had a good queen, and the colony where they had a bad brood pattern and a supposedly bad or failing queen, and we'll swap them. Okay? So what she did is went back then 21 days later and measured the brood pattern. She measured the brood pattern before and then after the swap. And guess what she found? This is in essence what these graphs show, but I won't explain those. I'll just give you the punchline. The supposed bad queen that had a bad brood pattern, put that into the good colony, her brood pattern improved. You put the good queen with a good brood pattern, put that into the bad colony, her brood pattern got worse. In other words, it was a colony level trait. right? It was not the queen per se, okay? So bad queens may not always be bad. Again, we're assessing the queens based on the colony. And so I think we need to rethink this. We need to be a little more suspect to it. Sometimes it is the queen's fault, but I think more often than not, we're finding that it's really not. And it's much more of a complicated way that we're assessing queen quality. So another queen event or another queen problem is kind of different from brood pattern, although kind of similar, where it's not just the spottiness of the brood, either due to the queen or due to disease or something else, but rather that she's not inseminating she's not fertilizing the eggs properly and becoming a drone layer, because unfertilized eggs become drones, right? So here's an example where you can have a lot of drones within the colony. But what's this? This is laying workers, right? So this is an easy one where you see these multiple eggs within the colony or within each cell. They're laid on the side. They're laid not all the way down on the bottom because the workers' butts are too small to reach all the way to the bottom. So this is a telltale sign, and boy, these things are impossible to requeen. Not impossible, but really, really difficult to requeen once they get to this stage. But a lot of times they'll raise these as drones, but they come out really runty small drones because they're in worker-sized cells, right? So you get a lot of small drones and drones in worker comb. So this is a problem, but it's not really the queen's fault because she hasn't been around for 2 weeks or more, right? So it's hard to say. That's kind of a colony level trait. What's this? Is that the same thing, laying workers?
Hygienic.
It's not hygienic. Somebody said it, drone layer. But there are different types of drone layers, okay? This is a picture of a drone layer because the queen never mated. In other words, the spermatheca was empty, okay? The spermatheca is fixed. It's a spherical organ, not as big as my hand, it's much smaller. But imagine this really small thing in the queen's abdomen, and its membrane is fixed. It's pretty rigid, actually. And virgin queens, it's completely filled with saline, with a fluid, right? So it's clear. Whereas when a queen gets fully mated, it fills up with the sperm, and it kind of takes this tannish, brownish, marbling type of look to it. And so virgin queens that are never able to go out and mate, or they don't find any drones or something, they don't get a full spermatheca. When they start laying, pretty much, every single egg that she lays is unfertilized, therefore it becomes a drone. Versus this, what's this? Same thing? It is the same thing, but it's for a different reason. This is a drone layer too. But in this case, her spermatheca is full, but all the sperm are dead, right? How do you tell the difference between those two? You can't, right? At least based on the brood pattern. You can know it by the timing, if it's kind of a young queen that should have just mated, but she's only laying blanks, then you know that it was an unmated queen. If it's an old queen, right, then it's more likely that, and she was laying a good brood pattern, but then all of a sudden it transitions over to nothing but drones, then you can say that either she ran out of sperm or it's a dead sperm, right? So again, not all drone layers are the same. Here's another thing that'll really blow your mind. Through our clinic and through working with beekeepers and doing a study, we tried to do a study last year where we wanted to make low quality drones by having drone layers lay drones in worker-sized cells. And so we make these puny little drones, we make normal drones, and we'd have that same continuum like we did for our queens, because now we're offering the same service that we do for queens, we do for drones. So you can send us drones and we can analyze those in our clinic. Well, in doing so, we were soliciting beekeepers to send us drone layers. Hey, whenever you find a drone layer, rather than pinching her head, send them to us and we will use them for this experiment, okay? Well, we had 17 queens, drone laying queens sent to us, all right? 40% of those were indeed laid only drones. 40% or 20%, sorry, laid drones, but then a couple days later started laying beautiful worker brood. The other 40%, drone layers shipped to us, they were perfect from the get-go. In other words, it's very possible that drone laying can be a recoverable condition. That's crazy, right? So we think that, oh, well, if a queen never mated, never got a full spermatheca, well, of course, she's only going to lay drones. She has nothing to fertilize it with. Or here's an old queen, she starts failing, laying drone brood. Well, clearly all the sperm is dead. Well, there's a third possibility that maybe something's happening either in the environment, like somehow the sperm is not fertilizing the egg because the wax comb is too polluted, or maybe there's a physical blockage of the spermathecal duct so that when the queen releases the sperm, it doesn't actually get onto the egg. Or maybe the queen has an infection like nosema that kind of blocks her up in some way, and so she's able to lay eggs, but she's still not able to fertilize it. But then when you cage her up and you ship her through the mail, she somehow kind of rids herself of this infection, and now she can fertilize eggs again. So this is, again, something where we shouldn't rush to judgment and think that these things are maybe permanent, right? This may be something where not all drone layers are hopeless. So again, we need to look into that to see how often does that happen. I mean, this is a very bad sample size, right? We need to do this at a lot higher numbers and see how frequent this really is. But at least the possibility that drone laying can be a recoverable condition, I think is kind of tantalizing. All right, so here's one last kind of queen event or problem, right, where the queens are no longer good and therefore want to be replaced by the workers. Now we all know that swarm cells always are on the bottoms of the brood frames, whereas supersedure cells are always in the middle of the brood frame, right? Because always is something that we use in beekeeping all the time, right? Not diagnostic by any means, but maybe 51% of the time this is true, but 49% it's not, I don't know. But looking for supersedure cells, even when the colony seems happy and the queen looks good, is something that we need to have a higher priority for, right? Like, I think things are going on and the bees are making these weird choices, or they're superseding when we would, not expect them to do so. You also have other problems where, they're trying to raise queens. You ever seen these really long kind of queen cells? You ever seen those? Sometimes that's built around a drone larva, and then they really don't know what to do with it. Or the larva can fall out of the royal jelly and kind of topple down, but instead of just getting rid of it, they kind of keep building the cell down. Weird things like that. Here's a cell, virgin queen emerged from it, right? Reading the comb is important. And then here's an assassinated queen. You have these virgin queens. This mouse. These virgin queens that are kind of roaming the comb, and rather than waiting for their sisters and rivals to emerge, they'll chew a hole in the side of the cell and sting her before she has a chance to fully mature, so that she has a better chance at taking over the colony. So you can see these kind of weird things too. But this is all after the fact, right? The colony has already decided to make the next generation of queens. There's already something wrong with the queens from their perspective. And we know, again, that low-quality queens form low-quality colonies. And because supersedure cells are often built around older worker larvae, they're of lower quality, and therefore supersedure queens are usually not as good queens and not as good colonies. So low-quality queens are, another thing, part of this study, you know, we formed colonies headed by high-quality queens and low-quality queens. And we're also measuring the supersedure rates, hoping and expecting that the low-quality queens were going to be perceived by the workers and superseded more readily. Nope. So what we consider low quality and all these measurable effects of queen quality, the workers don't seem to care. Yet, they're constantly trying to supersede what we consider high-quality queens. Go figure. So this is my current thinking, and this is very complicated, so I apologize ahead of time, but hopefully I can kind of walk you through this, because this is where my brain is right now, and where my thinking is, and where I hope our lab is going to be going in the future, and looking at kind of queen quality in the entire aggregate, and thinking of it not just at the queen level, but at the colony level as well. And so if we look at a queen's lifetime over time, and this is nonlinear, right? So here, this is just a matter of days, these are months, and you know, up to years. You know, queens are supposed to live 2 to 3 years or more, okay? But there's 4 stages impact a queen's quality from our perspective, and supposedly the bee's perspective. The first we've already talked about is the developmental phase, right? Where the queens are reared, and then they mate, and then they start laying eggs, right? That usually takes a couple weeks, that developmental phase. So we've studied that a lot. The second phase is what I call the establishment phase, which is once the queen starts laying eggs, you have 3 weeks from her starting to lay eggs to when the colony population turns over. Well, 3 weeks for her first offspring to emerge, and then probably another brood cycle or 2 before now the entire colony population is hers, right? Now, of course, in beekeeping, this transition phase from when the queen starts to lay eggs to being introduced to a colony, this is where we have this transportation and shipping and these other things that we do managerially, right? But anyway, that's a second phase, this establishment phase. After all of the offspring has turned over and all of the offspring in the colony is her worker daughters, this is what we call the survival phase, where she's fully accepted by her daughters and up until she becomes too old. Now this is where we as beekeepers want the queen, this is the maximum phase. We want to maximize this phase, right? To get as long a period of time out of her. We want to maximize her longevity. And again, it used to be that queens live 5 years, now they're barely living 1. So this is the phase that we really want to maximize here. But even the best queen that can live 5 or more years, she's eventually going to get old, right? And so that's the final phase that we call the replacement phase, where she starts to fail for some reason and eventually does get superseded and replaced by the workers. Everybody follow that? Is that okay? All right. So here are these 4 phases. And then when she is replaced, there is some probability that they're not going to requeen successfully, but hopefully they do. But that's a problem, is this unsuccessful requeening. So that's something that we need to look into, because sometimes they start to requeen and then they fail to do so, and then they become laying workers. And you're like, what the hell? But, you know, that happens. Now, if we just look at that insemination success of the queen, where, you know, when she's a virgin, her spermatheca is empty. When she mates, it gets full, or as full as it can be. But as she's laying eggs over the course of her lifetime, her sperm storage depletes, and eventually she'll run out, or at least she'll have fewer sperm than she started with, right? No argument there. The viability of the sperm is not always 100%, so that kind of concentrates or can accelerate when that happens, right? If the sperm viability is really low, she's going to run out of live sperm a lot faster. Right? So this is where I think the reproductive quality of the queen then interfaces and intersects with the workers. Because the workers are somehow judging the queen, right? And somehow collectively, we know that they can make amazing decisions about what nest site to go to, or which, you know, plants to forage on, or these other things. We know that they can make these collective decisions in these very smart ways. Well, they're making a decision of the queen. They're forming some sort of gestalt-like opinion of her, where at some point she's acceptable. Okay, we like the queen, she's laying eggs, everything. But then when she gets into this replacement phase, at some point she crosses some threshold. And they say, okay, she's not acceptable anymore. It's a binary choice, right? You either keep the queen you have, or you get a new one. It's like us. When we come up with an election, you either keep the dope that's in there, or you elect a new dope to go in there, right? That's our choice. It's a binary thing. So the workers must have some sort of means by which they're making these collective decisions. We have no idea what that is, but we're interested in trying to understand that collective decision-making process, because once they cross the threshold, then they try to replace her, right? So what happens is that under normal conditions, she lays for many, many years. Eventually, though, she crosses that threshold and they get superseded. But outside influences, or maybe inside influences inside the colony, are causing that a shift where that normal deceleration process happens sooner in the queen's lifetime, during the survival phase. Or, as I pointed out, even when you install packages and before the population turns over and becomes all of her offspring, they can cross that unacceptable threshold even during the establishment phase. So what are the factors that are driving this? And so what we hope to do, to go back to all of those different factors that influence these cues or characteristics or environments within the colony and from outside, they're probably interacting in very complicated ways, that all of these factors are affecting things that are affecting these different phases in different ways. So the take-home message here is that it's complicated, right? But the point is that putting all of these things together in a very quick way, right? Oh, brood viability, not good, bad queen. And so if we really want to drill down into this, I think we need to recognize that the things that we're judging, you know, the queens by are not always the queen's fault. And even at these different phases of the queen's lifetime, there may be different factors, or even the same factors, that are interacting in different ways. So we hope to tease all of that apart and kind of have more than just a concept, but hopefully more of kind of connecting some of these dots. But in the meantime, just realize that this idea of why good queens go bad, you know, we're blaming the queen a lot of the time, but I think most of the time it's either both the queen and the colony, or really just an environmental colony thing. More often than not, it's probably a rare thing that it's only the queen's fault. And so we need to think of the queen and her colony more in the collective at the colony level than really at the queen level. We need to get away from this idea that, oh, all we have to do is requeen and all our problems are solved. We need to be a little more introspective to our own management practices and to kind of really think more holistically that the queen's problems are really more a symptom rather than a cause. And with that, I'd be happy to take your questions. Thank you. Yeah. Mm-hmm.
You were mentioning on that last slide about the pheromones in the colony, and I was wondering, do you have any negative pheromones so that you can monitor if it's changing, perhaps as the queen's health or as the colony dynamics change?
Yeah, the question's about pheromones and if there's a way to measure those. So whatever this gestalt impression is that the workers have of the queen has to be mediated by some sort of communication, right? And pheromones are the main means by which bees communicate. And so my thinking is it's not necessarily just the queen pheromones that regulate mating and the retinue and inhibit queen rearing. That must have something to do with it. But my thinking is, just based on the paucity of literature that we have, is that really brood pheromones of my guess of what is potentially triggering a lot of this. That if the queen is not a good egg layer, then they're not making brood. Brood makes pheromones. So if the bees aren't smelling this brood pheromone, there must be something wrong with the queen, so let's get a new one, right? Your question was, do we have a way to measure this? The answer is yes, by smarter people than me, and it's really, really, really hard because it deals with very complicated chemistry. Now, Brad Metz, who's in our lab, he is one of the world's experts on brood pheromone. Oh my goodness. But it is incredibly difficult to study because it's so environmentally sensitive. So the answer is yes, but it's not easy. And I wish we had a field test for that or something, but we don't. Yeah?
We're getting rid of the queen in the swarm. We're not really getting free bees. We need to re-queen whenever we get.
Yeah, when you're capturing a swarm, and until we get Africanized bees in here, that's okay. We don't capture swarms anymore. But it's still probably a good idea to requeen that swarm when you can, because at the very least, you're getting an old queen. At the very worst, you're getting a nasty queen from your neighbors, right? Or somebody, or feral, right? So in order to control those genetics and make sure that you have, control over your domain, requeening that is recommended, yes. In the back.
So let's go back a little bit to your last talk on queens, but you hear a lot about how it's good to buy local queens or raise your own queens because they are genetically adapted to the environment. But what I'm getting from what you said is that based on the meta-national
Yep.
data that you've analyzed, that yes, it's good to buy local because you at least know who's raised them and that they're raising healthy bees, but it doesn't necessarily, like, buying Minnesota queens and raising them or using them in your North Carolina hive, it's not going to really make a difference, like,
Right.
if they're not actually adapted to cold climate.
Correct. Yeah. So I guess if I can summarize your question, here's a hand grenade, please pull the pin. You're correct that there is no locally adapted stock is a very charged thing. And Tom Seeley has been studying the Arnott Forest, and there are commercial queen producers that have large enough populations and isolated enough areas that they probably do have locally adapted, or at least better regionally adapted stock than others. But by and large, genetically, all of our bees are just one big population, and we do not have that as long as we're moving bees around. So stop moving bees around is the first and necessary starting condition in order to achieve locally adapted stock, right? But it takes generations and generations to demonstrate that they do better here than they do better anywhere else. And that is a much higher bar, right, to cross. So starting local queen rearing is a necessary first step in doing that. So that's why that's encouraged. It also negates, or at least minimizes, that transport step where there can be stressors during transport that can then affect the queen as well. If you buy local, you don't have to ship them from Hawaii, right? So I think there are multiple benefits to that. But genetically adapted to a local environment, they're all introduced to the new world, so we don't have that. The best that we can come is start and provide the conditions that will enable that to happen, but it hasn't happened yet because we're shipping bees all over the place. Yeah.
I had a hive lately turned mean all of a sudden. What I've been wondering about is when the queen mates with the drone in her spermatheca, does the sperm lay in layers?
Oh, that's a great
where
question.
She would all of a sudden start laying mean bees because she got with a bad boy, and then maybe that would clear up later?
Yeah, no, that's a great question. It was predicated by saying that you had a colony that was nice and then kind of turned mean. But the idea is that when queens mate with, say, a dozen drones, is the sperm layered in the spermatheca, so she goes through one drone where the workers are nice, and then another drone where the workers are not so nice, and vice versa. The answer is no, and that's been very well studied, that in the process of mating and in the entry of the sperm into the spermatheca, it's very well mixed. So at any given time, it's a Monte Carlo crapshoot of who fathers a given egg. So it's probably an environmental effect or something else that's happening, and not that she's going through one drone sperm at a time. She's using all of them all of the time. Yeah, that's an important point. Yeah.
I want to go back and ask you way back in this talk about speaking about the inferior queens, the inner caste, and that's caused by poor nutrition mostly. Or wrong nutrition at the wrong time. So that does produce poor quality.
It does,
colony.
yes.
Is she in fact though producing poor quality offspring? Because I mean, I guess by it being the food rather than her characteristics that caused her to be a poor queen. Could she in fact produce good workers and then maybe even produce a good queen that could have to flourish?
The question was about those studies that we did where we established colonies with high versus low quality queens. And the question was, do they make low quality workers? We actually didn't test that, but I'm kicking myself that we didn't. My guess is that low quality queens, right, I should say that the parsimonious explanation is that low quality queens make equally high quality workers as a high quality queen would, just less of them, because they can't lay as many eggs per unit time, they haven't mated as much, so on and so forth. But it's really intriguing of what you just proposed, which is there a maternal effect, not a genetic effect, but do low quality queens make low quality offspring because maybe they're not nourishing the eggs as much, which we have shown, right? So that's why I'm kicking myself that we didn't sample those workers and do some more finite analyses of them to see if the low quality colonies produced by low quality queens is a function of quantity of workers or quality of workers. Yeah, so that's still up in the air. That's something we should do.
That would, if in fact they were good quality workers
Yep.
coming from her, then the hive maybe wouldn't be doomed by one of these inner-caste queens.
Right.
They could actually produce a quality queen from a whole.
No, correct. And if you notice, it's not a huge effect, right? It's not like a night and day effect of a colony head by a high quality queen versus a low quality queen. But yes, if they make the third generation of queens, then they should be just as good, right? So yeah, all of that needs to be teased out, but it's a very intriguing possibility. You had your hand up too, ma'am?
In our area, they're starting to sell queens that they're calling
Saskatraz, yeah. Yeah, so Saskatraz queens are a line of queens up in Canada that they've been breeding for winter hardiness. They started with the Russian stock, I believe, mixed with Carnies. Is that right, Jennifer? Russians and Carnies. Yeah, she was paying attention, sure. I believe, well, since she can't say I'm wrong, I'm just going to go with it. But they've been selecting very heavily for winter hardiness, low mite control, or low mite levels, and other characteristics. So it's a new stock that is been commercially propagated, and we can get now. We've done some studies with them, not for the purposes of that, but it is a new thing that another option that we should try. Now, that said, getting back to that question before about the locally adapted stocks, here's a stock that was bred for winter hardiness. We don't have a winter compared to, right? Like zero winter compared to Canada. So if that is locally adapted, they're probably not a good choice down here, right? But try them. If you like them and they do well, then great, keep using them, but they're probably then not locally adapted to Canada, right? So again, I think a lot of this comes down to your management style and kind of what you're trying to get out of your bees, and try what you like. Yeah, in the back.
You're talking about everything basically being the same genetics. What about what Sue Goldie's doing? Producing new alleles, sex alleles. Good thing, bad thing?
Yeah, so the studies that we're looking at about the panmixis, again, that was all based on Italian stock and the genetic analyses that we had done in those series of studies. But different stocks, right, are going to have slightly different genetics. Again, there's more variation within a stock than there is across stocks, in the sense that because they can all interbreed, it's really hard to keep a pure stock here of the same species, right? But going over and getting germplasm from the old world and inseminating queens here is a way to introduce new alleles, right? So that would be different compared to the Italians, right? But I don't think it's kind of like night and day. There's still going to be a lot of overlap in those populations, right? Because again, everybody in North America is mating with each other. That sounded bad. Let me rephrase that. Because we're genetically moving these different types around, it's hard to keep those genetic lines pure. Great. Thank you again. Appreciate it.